Okay, so now we're moving on to Chapter 2. These are some of the comprehension questions from Chapter 2. There are a lot of these comprehension questions, and it's worth going over them and trying to come up with the answer yourself, looking in the book, using these questions as a guide to how you approach the material, as opposed to just sort of passively reading the chapter. In fact, if you just go to these questions first and then try to answer them, I think that's a good way to go about it. Most of these are pretty straightforward in terms of that you can get the answer from the solutions manual after you've tried to answer themselves, just to check and get some of the details. And so I don't feel it's necessary for me to go over all of them on video, but some of them are more important than others, and these three get at the heart of really what I want you to get out of this chapter. And so this chapter is all about meiosis. We've talked about how genes are inherited, and we've talked about how we've already gotten into how the chromosomes are inherited and how they account for how genes are inherited a little bit, but this chapter goes into it in some detail. And so first of all, what are the major genetic results of meiosis? And if you have two genes, you can certainly have more genes, but typically we're talking about two genes. You go through meiosis. And you go through meiosis 1 and meiosis 2, and you end up with gametes. And these gametes, if everything's happening according to Mendelian genetics, have these possible genotypes, and each of these is equally likely. So you can get 1, 1, 1, 1, or 1/4, 1/4, 1/4, 1/4. So we know that from a genetic standpoint, and what we mean by a genetic standpoint is that if you do a cross, and let's say we did a test cross, because the test cross is sort of the easiest way to see what's happening with the genes. And whoa, this pen is acting up today, so we might not be able to do it. But and then here you would get the big A. And you always get the little a from the other parent. But here we're going to get the little b from the parent on the left and a little b from the tester that's the parent on the right. Here we get a little a from the parent on the left, a little a from the tester, a big B from the parent on the left, and always a little b from the tester. And here we get little, 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 little. And so in this case, the phenotypes all have the same ratio as the gametes. And that is because of independent assortment. So the reason that we get, well, first of all, the reason that we get big A segregating from little a is segregation of alleles, and the reason that we can get a big A with a big B or a big A with a little b is independent assortment. So this chapter is all about how the chromosomes account for that. And so the question is, what are the products of meiosis, or what are the major genetic results of meiosis? And they're gametes that are like this. So I guess the official answer would be formation of gametes. And segregation of alleles, independent assortment, and also going from diploid to haploid. So that's an important part of meiosis. And so we could represent diploid is often called 2N, and this is N. And so the only time this happens is during meiosis 1. Meiosis 1. And so we'll look at the chromosomes and how that occurs. And so these are the major genetic results of meiosis. This independent assortment also gives you a lot of genetic variation. We started with, so we start to get multiple different kinds of things coming out from the crosses. And so I guess that's 14 and plus a little bit of extra info. 15 would be what are the processes unique to meiosis responsible for genetic variation. The big one is independent. The big one is independent assortment. So genetic variation. A lot of people like to say crossing over, and that's the other question, or the other answer to the question. But people like to forget about independent assortment. What's the other one? Crossing over. And so I like to even just not even worry a whole lot about crossing over until we get to whatever the chapter is on genetic mapping. Because crossing over is important, but I don't really think we're going to do much with crossing over until we get to mapping, because that's where it really comes into play. And so when do these occur? Well, it turns out that independent assortment, at what point during meiosis do they take place? That gets at the stages of meiosis, which is kind of important, but it's sort of a continual process. But really what is responsible for independent assortment and segregation of alleles is metaphase 1. And then, of course, and so I'm talking about how the chromosomes line up at metaphase 1. But once they line up at metaphase 1, you have to have anaphase so that the chromosomes go apart. And crossing over actually happens earlier. Crossing over happens in prophase, and actually pretty early in prophase. It's pretty interesting. Crossing over actually helps the chromosomes line up. So part of the process of prophase is chromosomes finding each other and then forming, they sort of get intertwined, and that brings the chromosomes together and helps them align. We can talk more about that. That's pretty cool. And so those are the processes. And then, so crossing over an independent assortment, don't forget about independent assortment. It's really important in generating genetic variation. List and describe the major differences between mitosis and meiosis. Include a description of how chromosomes align at metaphase of mitosis and meiosis. So the main differences are mitosis. Maybe we can put these in a little list here. Mitosis, meiosis. And mitosis is equational. Meaning, and this is kind of part of the same thing, you get identical daughter cells. So the whole point of mitosis is to start out with a cell and have it divide and come up and have the same kind of cell. And so if you start out with a diploid cell, you're going to get a diploid cell. In some cases, you can actually start out with a haploid cell. Some organisms are always haploid, or they have haploid parts of their life cycle, and they can divide by mitosis. And if they're haploid to begin with, they're haploid after mitosis, because they have to be identical. I like to throw in the little caveat that sometimes there can be mutations and stuff like that, so they may or may not be 100% identical, but for the most part they're identical. Meiosis is reductional. That's one of the most important things. Reductional. What does that mean? That means go from 2 N to 1 N. We reduce the amount of genetic material. That's important because this guy is going to form a gamete that we want to fertilize with another gamete And once we fertilize it, we can take this N and we can go back to 2N. And that's the sexual life cycle. You start out as a diploid, you make haploid gametes, and you make a zygote, and that zygote is diploid. That zygote then divides by mitosis and makes a whole bunch more diploid cells, which eventually makes a human, or whatever organism you're working with. It also, the daughter cells are different, so there's genetic variation. Because of the way chromosomes line up, you get half the chromosomes go to one cell, half the chromosomes go to the other cell, and which ones go to which cell is different every time you do meiosis. So you generate a lot of genetic variation. I'm sure there's more, but those are the only ones I care about. Meiosis reduces the number of chromosomes, and it generates a lot of genetic variation. How does it do that? Metaphase 1. What does that mean? So, big A, big B, little b, and chromosomes lining up. I'm going to draw them like this time. So, well, first I'll draw them like this. We'll come back to the cell cycle a little bit later, and then we'll draw another one here. So you can draw chromosomes like this, and there's going to be a locus on the chromosome that contains or carries the allele. So this is the A locus. That's the location. Locus just means location. That's the location on the chromosome where the A gene lives. Now you can have different alleles. So big A is one allele, little a is another allele. And then this chromosome, I'll put B down here because because I got more room down there. And put little b there. So this guy is heterozygous. This is what its chromosomes would look like. This is sort of what they look like. This would be before S phase. So we have to, in order to get them ready to divide by meiosis, it's a little bit hard to draw here, but the chromosomes end up looking, I should have put that A over there. Here, let's move this A out of the way. Get out of the way. All right. And then we can put it over here. And then we have DNA replication. And we have another A. So notice that big A go together, little a go together. These are, excuse me, these are homologous chromosomes. These are sister chromatids. And if we ignore crossing over, because it just is there to confuse us now, before crossing over occurs, sister chromatids are identical. Now so that's what we're going to say. Identical. They have to be. They just got copied. This got copied and made a copy of that. So DNA replication, copying that. DNA replication, copying that. It's going to have a little b on it. It's going to have it in the same spot. And it's going to have a big B in the same spot. And so you notice how they're. So meiosis 1, you always have to have two chromatids, because that's just how they are at meiosis 1. And so then we can show how they. So that's metaphase of meiosis. And so I'll just draw them again. I'll draw them a little bit differently this time. Here, this is a little easier to draw them this way. So there's a centromere. There's a sister chromatid. There's another chromatid. Here's another chromosome. And be careful when you're speaking and saying the word chromosome that you say chromosome and not chromosome. A lot of people say chromosome. I think sometimes sounds almost like I'm saying chromosome, but you have to say chromosome. And say when you have a gene at a particular spot that's a locus, if you're talking about multiple locuses, the word is loci. So plural of locus is loci, just like the plural of focus is foci. And we can put these up here. It doesn't really matter where these go. So there's meiosis. Looks like that. Because they look like that, A is segregated away from each other, B is segregated away from each other. See if I can, and I think we can fit them all on here. So we're going to put mitosis over here. I don't know why this pen's not working so good today. Hopefully it's nothing. I don't have to buy a new pen already. So mitosis we'll draw in a minute. But I first want to draw that there's two different ways that the chromosomes can align during meiosis. Yeah, you guys are probably getting bored of seeing this because I've drawn it a million times, but that's the first question on the exam is going to be for you guys to do this, so you might as well see it a bunch of times. So I'm going to add, this is the answer to the first question on the exam, or maybe not the first, but certainly a question on the exam. Draw the chromosomes as they line up during meiosis. We can't have a B there, but we can easily have an A there. We can turn that into a little B. Easy enough. Oh. Alright, you guys get it. Big B over here. Keep making that little circle there. Big B. Big B. Alright, so now when these things divide and we go through meiosis and we go through both meioses, these are the kinds of things that can be made. We won't worry about the fact that there's two divisions at this point. We'll just sort of jump ahead and show. So this A goes with this B, and this little a goes with this big B. And because of the way so we have to actually go to two divisions because there's meiosis 2, but what you end up with is 2 of these and 2 of these and 2 of these and 2 of these, and we just break that down to 1 to 1 to 1 to 1. So that's the two ways that they can line up during meiosis. These would be in two different cells that are going through meiosis, and then in mitosis. So the way you say, so the way I like to think of it is in meiosis they line up in pairs. In mitosis they line up single file. So when I draw this, it's getting more and more abstract as I go, but this is a centromere. These are sister chromatids, and they just line up in a row. And it doesn't matter what you put where, which chromosome which is which. They just sort of, however you want to put them in here, that's what they are. Alright, so that's how they line up during mitosis. And so when they divide, this cell over here is going to get one of each. It's going to get a big A. It's going to get a little B. The little A there. It's going to get a big B. It's going to get a little B. This other cell is going to get a big A. It's going to get a little A. It's going to get a big B. And it's going to get a little B. So there's a cell. There's a cell. And remember, we started out, in both these cases, we started out with this. Big A, big B, little b. So here, we end up with big A, big B, little b, because they're identical. But here, we end up with half the number of genes, and we end up with different combinations. Big A, big B, big A, little b. So independent assortment. All right, so those are the big sort of overview questions, and then some of the other questions in this chapter are designed to make you think a little bit and to sort of knowing what's going on with the chromosomes, asking you some questions that make you sort of apply that information. So for now, so that's basically it. Actually, now that I think about it, there's a couple more things I'll point out about meiosis. And keep in mind that meiosis, we don't really care about mitosis when we're talking about the genetics and genetic crosses. Mitosis is important and interesting for lots of reasons, But when we're talking about how genes are inherited during sexual reproduction, which is mostly what genetics is right now, we're talking about meiosis. But I want to talk about chromosome numbers. And so I think we will give myself a little more room here. So let's see if I can draw some slightly better chromosomes here. So they line up in pairs. We're going to put both divisions in here. So here's one chromosome, here's the other chromosome. Maybe I don't even need to put the genes on them at this point. Put another chromosome, they're lining up in pairs. They're homologous chromosomes. So they line up in pairs. In meiosis 1, they divide. Cells divide and the pairs segregate away from each other. So this is meiosis 1. And so, when the chromosomes are lined up like this, then it's metaphase of meiosis 1, and then you go through anaphase of meiosis 1, and telophase and cytokinesis and everything, and you end up with these cells. And that is the reductional division. So meiosis 1 is the reductional division. It's the only reductional division that exists in the universe as far as we know. So that's the whole point of meiosis, reductional division. And then you get another bonus division down here. So this is meiosis 2. And you get metaphase 2, so they'd kind of look like this, like they do up here in metaphase 2. And you can kind of think of metaphase 2 as being like mitosis, because they then separate and you only get one chromatid from each chromosome, and you get one chromatid from each chromosome over here. So this is what happens in meiosis 2. So this is actually what your gametes look like if we start looking at all the chromosomes and looking at the cell and looking at both divisions. And here we go. And so these guys came from the cell. You could put genes on there. Maybe we could change some colors here. Put some genes on there. And in this particular case, let's say we'll just put one on there just to keep it simple for now. We'll line them up like this. There's two different ways they can line up. If they line up like this, now we're going to have an A on each chromatid. Now the first thing I want to talk about is, so what is the diploid number of this cell? So 2n is the diploid number of this cell, and the answer is 4. There's 4 chromosomes. So count your centromeres. There's 1 centromere, 2 centromeres, 3 centromeres, 4 centromeres. That means there's 4 chromosomes. After meiosis 1, which is reductional, now you have 2 chromosomes per cell. It gets confusing because a chromosome is this is called a chromosome when it has 2 chromatids, and this is called a chromosome when it has 1 chromatid. So it's just, you just have to keep that in mind. So at this point, this would have gotten a big A and a little b, and this would also have a big A and a little b, because those are sister chromatids, so they have to be identical. And then when you get this division, you get big A, little b. So genetically, we call this big A, little b, even though there's 2 big A's and 2 little b's. We don't really think about that, and so we're worried about what's going to end up in the gamete. And we get 2 of them, because that's just the way it goes. And then this is, if we follow this through, we'll get a little more abstract here. We got big A and little b are going together. So these guys get a little a, big B, little a, big B. So what we get out of this are one type of gamete. We actually get two cells that have this genotype, and then two cells that look like this. But if these chromosomes had lined up the other way, so that big A lined up with big B, so if they had lined up the other way, so that way up here at the beginning, if big A and big B were together, then the gametes would be big A, big B, little a, little b. So that's how you get the other gametes, if these had lined up the other way, which they do half the time. But in this particular meiosis, we get these kinds of things. So just again, look at the number of chromosomes. We got 4 chromosomes to begin with, and that you would call that 2N in terms of haploid or diploid. And each of these cells has 2 chromosomes, and then each of these has 2 chromosomes. So that's sometimes confusing. So we've got 2 chromosomes, 4 chromosomes total up here, 2 here, 2 here. So this first division is reductional, and then the second division is equational, meaning it's equal. And just like mitosis was equational, meiosis 2 is also equational, because we start out with 2 chromosomes and we get 2 chromosomes out of it. Even though it looks like there's more stuff up here than there is down here, this is 2 chromosomes with 2 chromatids each, and this is 2 chromosomes with 1 chromatid each. And so we go 4, this is diploid, this is haploid, this is haploid. This has 4 chromosomes, 2 chromosomes, 2 chromosomes. When you go from 4 to 2, you're reducing the number of chromosomes in half, so that's why we call it reductional. Then you go 2 to 2, and that's equational. It's just there to confuse the issue. There's no major effect on anything. it's just we just talk about it because it actually happens, and so we have to talk about it. All right, so that's basically the fundamentals of how chromosomes are involved in meiosis and how it differs from mitosis. And so now we can go on to the more applied questions and think more about those.